In the first part of this program, we learned how body cells reproduce themselves by mitosis, so that the new cells contain the same diploid or double number of chromosomes found in the parent cell. Now we will learn how certain diploid germ cells, found only in the ovaries and testes, undergo a process similar to mitosis called meiosis that results in the production of sex cells that have only half the number of chromosomes found in the parent cells. In the case of human beings, the diploid body cells all have 46 chromosomes, whereas the sex cells, the sperm and egg, have only 23 chromosomes. This condition, where paired chromosomes are absent, is called the haploid condition of a cell. When two haploid cells, sperm and egg, join together for fertilization, a new diploid cell is formed that will then undergo mitosis over and over again, resulting in the creation of a brand new individual. Meiosis is similar to mitosis in many ways, but in other ways it is quite different. Now let's take a closer look at some of the details of this fascinating process. The earliest part of the first stage of meiosis, called prophase I, begins just as it does in mitosis. Chromosomes form from chromatin. These chromosomes contain DNA replicated during the preceding interphase. As prophase I progresses, two things happen in the germ cells that didn't occur during prophase in mitosis. First, the identical or homologous chromosomes form pairs with one another, so that the tall chromosomes find their mates, and short chromosomes do likewise. This pairing of homologous chromosomes during the first prophase of meiosis is defined as synapsis. The second difference between prophase I of meiosis and the one we saw in mitosis is that parts of one paired chromosome can actually be exchanged with parts of its opposite pair. This process, called crossing over, is defined as the exchange of chromosomal material during synapsis and occurs only during the first prophase of meiosis. During the next stage of meiosis, called metaphase I, the centromeres of the homologous chromosomes line up on the equator of the cell. In contrast, during mitosis, the centromeres of the homologous chromosomes align themselves in a completely independent fashion. During the first stage of meiosis, called anaphase I, the homologous chromosomes, each made up of two sister chromatids, move apart, centromeres intact, one toward each mitotic center. In contrast, during mitosis, the centromeres separate during anaphase, and the sister chromatids move apart as new daughter chromosomes. During the fourth stage of meiosis, called telophase I, each new cell contains just one of each homologous chromosome, and each of these chromosomes still consists of two sister chromatids. The fifth stage of meiosis, called prophase II, follows right on the heels of telophase I, without an interphase stage in between them, as happens in mitosis. This means the DNA of the chromosomes in the prophase II cells has not been replicated. And this explains why the chromosomes of prophase II cells have not sprouted any new sister chromatids. From now on out, meiosis follows the same stages as mitosis. During the sixth stage of meiosis, called metaphase II, the chromosomes line up on the equator of the cell. And now the centromeres split apart. And during the seventh stage of meiosis, called anaphase II, the centromeres separate for the first time. And now the sister chromatids become daughter chromosomes as they move toward the opposite poles of the cell. Finally, during the last stage of meiosis, called telophase II, new nuclear membranes form. And as the chromosomes change back into chromatin, we can see that 4 new sex cells have been produced from the single original diploid germ cell. But since the DNA was only replicated once throughout a series of 2 cytoplasmic divisions, the final number of chromosomes in the sex cells is only half the number present in the diploid parent cell. In other words, the sex cells are haploid. Now, when 2 haploid sex cells, sperm and egg, join together, the fertilized egg will contain the same diploid number of chromosomes found in any body cell. Meiosis is very important. It enables all organisms that reproduce sexually, that is, require both male and female parents, to do so without continually multiplying the amount of DNA in the nucleus. For without meiosis, the human diploid number of 46 chromosomes would reach 368 in just 3 generations, thus creating a very messy state inside third-generation cells. Meiosis also provides an effective mechanism whereby the DNA of the two parents may be resorted and recombined in new ways. And for this reason, no two human beings are exactly the same. Meiosis assures that each new generation will be unique and will always have new possibilities. The result is that each generation will always have its own great athletes, scientists, artists, musicians, and inventors. And as strange as it seems, meiosis also provides a physical bridge between successive generations, as parents pass along a tiny amount of their own substance, their own DNA, to their children. As a result of meiosis, sex cells are formed that have the number of chromosomes of body cells. True or false? Pairs of identical chromosomes are also called homologous chromosomes. True or false? Normal body cells are diploid because they possess one of each different chromosome. True or false? During meiosis, the DNA is replicated twice. True or false? In plant and animal cells, meiosis occurs only in germ cells.